Hi, I'm Travis Stiles. I'm the CEO of Novoron Bioscience, and we do drug development against a family of proteins known as the LDL receptor family. And this is a large family of proteins that are involved in a diverse array of important biology, but they've also been implicated in a lot of important diseases. But because of some misconceptions regarding the overlapping structure and function of these proteins, it's been long thought that they might not be very good drug targets. But Novoron's developed the tools and the depth of expertise to overcome these limitations and to be able to exploit the therapeutic potential in this space. What I'm going to talk about today is probably the most exciting example of what our approach can do, and that's in how we might be able to add years to decades of quality of life to patients that suffer from tauopathies such as Alzheimer's disease. And what I want to highlight before I get too much further into the LDL receptor family is kind of the state of the field, the state of the therapeutic field in Alzheimer's. And what I want to highlight here is that up until recently, we were really limited to symptomatic therapies, which are really useful early in disease, but over about 5 years, they start to lose their effect. And now we're really fortunate that we now have the very first ever disease-modifying therapies for Alzheimer's in the form of these anti-amyloid antibodies. But what I think is really important to highlight here is that even in the most stratified and patient-responsive populations, the practical benefit of these drugs is really only about 10 to 13 months of extended independent living or disease delay. And I think that we can all agree that even though you can't take away from an additional year of independent living or of extended life, but I think we can do better, and I think most people would agree with that. So how might we go about that? Well, one of the really interesting things over the course of the last few decades has been that increasing evidence has shown that the other half of Alzheimer's pathology, which is the tau tangles, which is as opposed to amyloid plaques, are actually a far more potent driver of disease than the amyloid proteins are. And I think it's really important that based on this to really understand a couple key differences between amyloid and tau. One, amyloid is an extracellular accumulation problem, where over time the amyloid will accumulate and disperse in the brain. But tau is actually a really important protein that acts as a microtubule stabilizer within neurons, and under normal circumstances serves a very important physiological role. But if it's overexpressed or dysregulated, it can form these aggregates, and those aggregates spread very differently throughout the brain. Unlike the amyloid, which kind of just disperses, tau actually will start in very specific areas and then will spread in very predictable ways through neuronal tracts. And it does that by actually traveling across synapses. And this is a very important fact to keep in mind. So, as I mentioned, the fact that tau has become more and more obvious to be a greater driver of disease, of course, there's been more drug development in this space, trying to target tau and exploit the therapeutic potential. And this is just an example of some really common approaches that are being taken in the clinic today. But what I want to highlight is the majority of these approaches are actually recycled from other areas. And these 4 here are actually approaches that we've already tried taking with amyloid. And as we know, only one approach has really panned out in that space in the form of the amyloid antibodies. And now you can imagine that even under the most favorable terms, if we were to extrapolate the antibody success in amyloid to tau, there would be an additional layer of difficulty where amyloid extracellular is going to be accessible to the antibodies, but getting inside the neuron is going to be another layer of difficulty. So I would argue that for how important tau obviously is to disease, maybe recycled approaches aren't the most efficient way to go about targeting this. And perhaps we should do something a little bit more bespoke for tau, something tailored to its actual physiology. And that's where I think that this synaptic spread and the spread of tau throughout the brain really comes into play. As in order for the dissemination through the brain to happen the way it does, neurons have to actually hand off tau across synapses from one neuron to another. And that's why it spreads across neuronal tracts, because you need connectivity in order for the tau to spread. And so if you were able to actually get into the synapse and block that neuron to neuron transfer, you could geographically restrict the tau spread and keep it into the defined regions wherever it is and not allow it to spread through the brain further. Now, this is not a Novoron idea. This is something that's been postulated in the field for quite a while, since we very first started characterizing that tau spread happens in such a distinct pattern as the way it does. What hasn't been necessarily known is how much that would matter for disease. And that's where I think that some of the more recent findings are really exciting. This is an example of 2 longitudinal studies of Alzheimer's patients who are genetically predisposed to early and aggressive disease. There's separate studies with separate genetic backgrounds, But in both cohorts, these patients are expected to get disease between the ages of 45 and 55. And the really exciting thing, or the interesting thing about longitudinal studies is, as you know the predicted disease course, when you find patients that deviate from this, either having later onset disease or earlier, you can look at their lifestyle, you can look at their genes, and you can try to find what might explain those deviations. But every once in a while, rarely, but every once in a while, you'll actually find patients that have exceptional disease resilience. So instead of a couple of years or months here or there, actually decades, or even demonstrating near immunity to cognitive decline despite having this predisposition. And what's really interesting is when you were looking at these being the symptomatic patients, the patients with cognitive decline, compared to the disease resilient patients on both sides. When you look at the amyloid burden, which is the top row, the amyloid burden in these resilient patients is actually significantly higher than their sick counterparts, which is a really interesting finding to see that the patients without cognitive decline in these cohorts actually have more amyloid. But what you find on the other side with the tau is, even though the tau pathology originates in the same way that you would expect for these specific patients, it stays restricted in this occipital lobe and doesn't spread through the brain, unlike in the symptomatic patients where this tau spread is robust. And this is really interesting because even though these are extreme examples, and even though some of these exceptional disease resilient patients have identified genetic changes or mutations that can mechanistically explain the protection benefits that we see, over the course of many of these resilient patients, what you find is the commonality isn't any specific gene mutation or lifestyle change. In fact, many times they're extremely different. The commonality becomes the restriction of tau spread. And the reason that that's so important is because, unlike the amyloid approach, which is talking months to maybe a year of benefit, in these patients we're seeing decades of resistance to cognitive decline. And so it stands to reason that if we were able to pharmacologically recapitulate this geographical restriction of tau spread, we could literally give decades of extended independence and quality of life to these patients. So how might we go about pharmacologically trying to replicate this effect? That's where LRP1 comes in. And LRP1 is one of the receptors we work with quite a bit. And back in 2020, one of our collaborators published a paper with the first author being another one of our collaborators, Dr. Jenny Rausch, with the lead author, or the last author being Dr. Ken Kosick. They published that if you take a virus that expresses human tau and you inject it into a very specific part of the brain, in normal mouse brains, that tau would eventually spread throughout the brain. But if you took away LRP1 specifically from neurons, and it's important to know that this was just a neuron knockout, there wasn't glial effects. Where the originating nerves expressed that tau started, it did not spread throughout the rest of the tissue. And so this is really exciting, and this is the quantification here. This is really exciting. So hopefully I've convinced you that, tau is a really important therapeutic target, but tau spread is a very important potential mechanism by which we can target tau. And that LRP1 is a really exciting therapeutic target that we can go after to accomplish this. So what do? Well, it's really important to note that LRP1 is a very large member of this larger family that I mentioned before. And if you look at this family, what you can note is they have very similar structure and overlapping functions. But because they're involved in such important biology, you have to tailor any therapeutic that you target here has to be able to disrupt the disease relevant interaction specifically, but leave the rest of the biology intact. And you can imagine how that might seem very difficult when these things look so similar. But what Novoron has done is we have built up a toolkit and a depth of expertise that allows us to overcome these limitations and be able to do things like very, very precise epitope determination for where ligands need to bind. And if those things are distinct enough from where other ligands need to bind, we can target that therapeutically to treat disease. And we've created independent models, structural insights, all sorts of screens and tools that allow us to be uniquely capable in this space of trying to exploit the therapeutic potential of these proteins. And importantly, when we developed a tool compound in-house that blocks LRP1, we were able to, across 2 models of tau spread, show that we can actually pharmacologically restrict the spread of tau. This, importantly, is a human iPS neuron model of tau spread, independent of seeding, and because seeding is something that people often use as a surrogate for spread, which we think is a little bit of cheating, and endogenous spread is actually more important. But in both this model and in the model I just showed you recently, where in the mouse model, where they used the knockout, we can actually use our molecule to restrict the spread of tau, which has two main proofs of concept here. One, it further validates the therapeutic target, but probably more importantly, it also shows that this receptor is druggable and that we can exploit it therapeutically. So building on that, and there's many different ways we can go about these problems with the tools that we have. But what was really exciting with this tauopathy specific program is we were able to determine when we interrogated the interface specific for tau that this was a very unique binding pocket that required a tertiary structure that deviated from some of the more standard interactions that have been characterized in this space. And that led to us doing some structural modeling, both in terms of like 3D modeling in a computer-generated system, but also needing to generate novel constructs of the protein to use in our biochemical assays that could recapitulate the tertiary structure that were very different from the canonical structures that are constructs that people use in this field. These constructs are so common in the field, you can actually buy them commercially because these are the dogmatic tools that people use. So we used our 3D modeling and we put it into an in silico screen to try to see if we could use that to predict molecules that might be able to disrupt this LRP1 interaction. And what was really interesting is as we got those molecules, we tested them both in our proprietary construct and in the canonical version as well. And what we found was when we tested the molecules in our construct, we saw this nice spectrum of activity, with the third pocket here that we were exploring being really, really exciting. But when we tested those same exact molecules using the canonical construct, we saw almost no activity. And what's important to note is that we then validated the activity of these molecules in orthogonal assays, some of not just biochemical, but also in cell-based systems, which means they are real. So this was really the first example that really proved our competitive advantage in this space. One, we're able to predict molecules that have activity based on our modeling. Two, we were able to create novel constructs, previously undiscovered constructs that were more capable of actually screening for the true interaction between LRP1 and tau. And that was the beginning of identifying these hits that led to a MedChem campaign. And without getting too deep into chemistry, where we really have made some big progress is we've been able to demonstrate feasibility in a small molecule program, both in terms of potency and in terms of the selectivity that can block the binding of tau but leave the other important biology intact. And this is really the kind of the holy grail of being able to exploit LRP1 therapeutically. Now, I want to leave you with three main points. One, tau is a really exciting therapeutic target, but tau spread is an incredibly unique opportunity to add decades, potentially decades of life, of quality life for patients with tauopathies such as Alzheimer's disease. Two, Novoron is uniquely positioned to exploit the therapeutic potential of LRP1 to accomplish just that. And three, success in this space really has far-reaching implications for other diseases that are related to members of this family that we could then potentially treat as well. And this is some of the larger opportunity, but our pipeline really demonstrates where we think the near-term horizon really lies for us. And so I appreciate your time. Thank you for listening, and hope to see you soon.